Angiogenesis is the process by which new blood vessels are formed from pre-existing capillaries. This process is normally very tightly regulated and does not occur except during embryonic development and repair processes. However, this regulation is compromised and contributes to pathogenesis of a number of diseases, including retinopathy of prematurity, proliferative diabetic retinopathy, and age-related macular degenerations, which are the major causes of blindness in humans. And some other diseases that more people are familiar with are cancer and arthritis, which also have this angiogenic component. And this led to a hypothesis that targeting this process may have a clinical relevance in terms of treatment condition. However, I believe there is a great gap in our understanding of how this process is normally regulated and what happens under these various pathological conditions where the new blood vessels grow. And that's really the focus of my research, and I believe without understanding how these normal regulatory pathways are functioning normally, it makes it very difficult to be able to develop strategies to interfere with this process and inhibit it. And this is really our ultimate goal in my lab, is to, again, to better understand how these normal processes are regulated and what happens under these pathological conditions that lead to the growth of these unwanted new vessels. Retina has two major circulation: the choroidal vasculature, which nourishes the back of the retina or the outer retina where the photoreceptors are. These are the cells that absorb the light and transmit the signal to the brain, and then you can see. And this is the circulation that is affected during age-related macular degeneration, especially the wet form of the disease, which has this new vessel growth into the retina. And the other circulation that nourishes the retina is the retinal vasculature, and this is the vasculature that is affected during diabetes and also in retinopathy of prematurity, which results in growth of these new blood vessels into the vitreous where they leak and cause problems. Normally, these vasculature are tightly regulated and kept on a very restricted pattern where these vessels only grow at certain places and not others. This issue has really fascinated many investigators, including our lab, that how are these restricted patterns are established. And this led to a hypothesis years ago that there must be some factors at some of these areas that normally prevent the growth of blood vessels to these areas, such as in the vitreous. For example, the retinal vessels never grow into the vitreous unless you have diabetes or if you are exposed to high oxygen in retinopathy of prematurity. That compromises this defense or this line of defense, and then these vessels grow into the abnormal areas. And now there is actually a growing list of some of these factors that are shown to be responsible for this type of regulation. Although initially the major focus of the many studies was on factors that promote angiogenesis, enhance the growth of new blood vessels, it is now appreciated that some of these factors that have anti-angiogenic activity play just as important role. And that's these factors, the factors that my lab has been interested in trying to understand how they normally participate in maintaining this ocular vascular homeostasis, and what happened to these factors under these various diseases that lead to growth of new blood vessels. My laboratory was actually the first laboratory to show that thrombospondin 1, which is one of the first most potent endogenous inhibitor of angiogenesis that has been identified, and we showed that it's present in vitreous and aqueous humor samples from eyes of different species that we looked at. And most interestingly, we showed that in animals that have very high level of this protein in their eyes, when they made diabetic, this protein goes away, suggesting that now the brake has been removed and now the tissue is susceptible to growing these new and unwanted vessels. We have now shown that this also occurs in human samples that we have collected from patients with and without diabetes that are undergoing surgery for their eye issues, and we have looked and we've shown that this protein is significantly decreased in patients that have diabetic retinopathy. And again, to provide further evidence, we have used some sophisticated genetic models in animal models that we use mainly in mouse, and we have shown that we can specifically downregulate or decrease the level of these proteins, mainly or more specifically in the vasculature of the retina, and show that when you do that, that exacerbates the pathogenesis of diabetic retinopathy in these animals. So our study clearly shows that thrombosponin 1, one of these first inhibitors identified, plays a critical role in maintaining ocular vascular homeostasis. In terms of utilizing that in terms of therapeutic, we and many other laboratories have been interested in developing and using short peptides that mimic the activity of this large protein, i.e. to inhibit neovascularization. And there is such a peptide has been developed, and it's been tested in a variety of preclinical animal model, which is very effective in inhibiting tumor growth. Again, tumor also needs angiogenesis to grow. And we recently have shown that this peptide is very potent inhibitor of choroidal neovascularization in preclinical animal model of AMD that is used in my lab. And most interestingly, recently we have assembled a collaborative team of researchers from Northwestern University and University of Nebraska, and here to further develop a newer version of these peptides that are more efficacious and also use different ways of delivery to further enhance the utility of this molecule for treatment of various eye diseases with neovascular component. Another area that my lab has been striding to make advances is developing ways to detect early changes that occurs during diabetes that leads to initiation and progression of diabetic retinopathy. Because currently, by the time when the ophthalmologist detect changes in your retinal vasculature, if you have diabetes, it's already too late. There are many changes that happen prior to the detection by an ophthalmologist that cannot be visualized or detected non-invasively. So again, thanks to the UWI Research Institute, which has been trying to cultivate collaborative work among different faculties with different background and research interest in vision, we actually put a team of investigators from Department of Mathematics, from biochemistry, and from biomedical engineering to develop a line of research which addresses this question. And we actually received a $3.1 million grant from National Eye Institute to look for some of these early changes that can be used to non-invasively detect early diabetic retinopathy changes. It is now pretty clear that collaborative research is much powerful and is capable of addressing major fundamental questions in research much better than a single laboratory can. And this has really been the mission of the Eye Research Institute, and the support from Retina Research Foundation and Dr. Alice McPherson has really been instrumental to promote and nurture the development of the Eye Institute and further enhance our effort in finding and improving our understanding of some of these issues and problems that leads to blindness. In a personal level, I have, as a recipient of the Dr. Alice McPherson and Retinal Research Foundation chair, I have received significant support from the Retinal Research Foundation. That's been instrumental in advancing our research in a number of ways and allowing me to develop a research program that is one of a kind. We were first to cultivate various retinal vascular cell type and choroidal vasculature from wild type and transgenic mice. These cells have been very instrumental in advancing our understanding and effort to get a better idea of how to prevent neovascularization. In addition, we have also provided many of these cell types that are very useful and precious tools for research to many laboratories in the US as well as other countries to use these cells in their effort. So for that, I am very grateful from the Retinal Research Foundation and Dr. Alice McPherson's support of this initiative at University of Wisconsin and my work personally in my work.